T.H. Chan School of Public Health and the Duke Molecular Physiology Institute. In the study, published on October 26, 2017, the team shed light on cells' declining ability to process energy over time, leading to aging and age-related diseases such as Alzheimer's and Parkinson's disease. Mitochondria, the energy-producing structures in cells, exist in networks that dramatically change shape according to energy demand. Their capacity to do so declines with age, but the impact this has on metabolism and cellular function was previously unclear. During the study, the researchers showed a link between dynamic changes in the shapes of mitochondrial networks and longevity. This was shown through the use of C. elegans, or nematode worms, which live just 2 weeks and thus enable the study of aging in real time. Mitochondrial networks inside cells typically toggle between fused and fragmented states, which affect how the organelles process energy as seen here. This is the fragmented state and the fused state. The researchers found that restricting the worm's diet through the genetic manipulation of an energy-sensing protein called AMP-activated protein kinase maintained the mitochondrial networks in a fused or youthful state. Therefore, the C. elegans with a restricted diet live longer than worms with an unrestricted diet due to optimal connections with their mitochondria. The same was true for those worms who AMP-activated protein kinase was genetically blocked, mimicking dietary restriction. Critical to these youthful networks that increase lifespan, the researchers found, were organelles called peroxisomes, which contribute to the oxidation of fatty acids inside the cells. The results support the researchers' hypothesis that periodic fasting and dietary restrictions promote mitochondrial homeostasis and plasticity and can be targeted to provide healthy aging and health and life-extending benefits. Heather Weir, a lead author of the study, explained the basis behind their research was from the idea that low-energy conditions such as dietary restriction and intermediate fasting have previously been shown to promote healthy aging, and understanding why this is the case is a crucial step towards being able to harness the benefits therapeutically. At the conclusion of the study, Weir is optimistic that the findings may open up new avenues in the search for therapeutic strategies that will reduce our likelihood of developing age-related diseases as we get older. Next steps for the researchers include testing the role of mitochondrial networks in the effect of fasting in mammals, and whether defects in mitochondrial flexibility might explain the association between obesity and increased risk for age-related diseases. And now back to Haley.
Thank you, Katie. Crowd vocal learning induces vocal dialects in bats. Playback of conspecific shapes, fundamental frequency usage by pups. Fruit bats learn vocal dialects not only from their mothers, but the entire population. According to a controlled study that manipulated the frequency of calls the pups were exposed to. One characteristic that sets humans apart from other animals is our ability to conversate and how we learn to conversate. However, a new study proves that fruit bats learn to communicate by social vocal learning. Social vocal learning is when an organism learns communication by being exposed to communication in a population. The fruit bats were tested in 3 different groups to observe if crowd vocal learning creates the bat's own dialect. When the pups were exposed to their mother's dialect, they recognized their mother's vocals. In addition, the pups were exposed to manipulated vocal frequencies from their natural roots. The results of this study showed that the pups had distinct dialects similar to what they were exposed to from the ages from 6 months and onward. Other animals can have genetic or ecological differences in dialect rather than learning differences, which separates them from humans. But for the fruit bat pups, each group was able to produce different dialects that were based on what they heard around them, which is similar to humans. And now over to Dylan.
Thank you, Haley, for your riveting presentation. My name's Don Kamani, and we're here at JBNN headquarters talking about how the time of day can affect how long it takes for your wounds to heal. So basically, it turns out that when you get hurt, affects how long it takes for you to heal. So the process of healing occurs underneath the skin itself. So, and this is affected by an internal clock that is akin to the circadian rhythm that affects how long you sleep and when you sleep, everything like that. But scientists still haven't been able to find out how long it takes for each cell or how each cell keeps the time and figures out when to start healing and everything. So fibroblasts are the skin cells underneath the epidermis, and they're mixed in with the dermis, and they kind of, so when a cut happens, it'll come probably right down the middle, and then the fibroblasts will come in right after the blood clots up. It'll come in along the edge of the wound, and then you'll see the skin start to heal after a period of time. So basically, like I said, the blood clots up, and that's called the hemostasis portion of healing. And then the fibroblasts come in along the edge of the wound, and they just create a new foundation for a layer of new skin to form. And after that, the skin just takes time to heal, and it's just, over time, it'll become just like new skin. And so basically, the amount of time that happens because of this, the amount of time that it takes for it to heal is affected by when you're wounded. So during the day, if your wound happens during the day, the wound will heal 60% faster than if it occurs at night. And scientists also found from the International Burn Injury Database that on average, nighttime burns took 11 days longer to heal than burns that occurred during the day. So like if you put your hand on a stove during the day, it'll heal faster than if you did that at night. I don't know why you would do that, but just as an example. And yeah, that's all, folks. So.
A recent experiment from Israel has found that stem cells could potentially cure paraplegia in humans. In this study, rats had their spinal cords cut and severed, which made them paralyzed. Scientists then modified human stem cells from the membrane line in the mouth, which they delivered into the rats. There were 4 groups of paralyzed rats, 3 given different types of stem cell treatment, and 1 was given no treatment. Independent observers then judged the behavior of the rats and how they recovered from their spinal cord transection. These observers were blind, meaning they did not know which rat got the stem cell treatment and which ones did not. After 5 weeks, when the test was over, the researchers did a pinch test to quantitatively measure the impact the stem cells had on the rats. If the rat responded in any way to being pinched in a certain area, then it was considered positive, and there's no reaction, the result would be negative. 5 of the 12 rats that were given stem cell treatment recovered and gained the ability to walk again, making the success rate of the best treatment 42%. This may not seem very impressive, but this type of paralysis is practically untreatable. Consequently, the rats that were not given any treatment did not recover at all. The reason for the rat rate being so low was because there is much unknown about both stem cells and spinal cord injuries. And due to the lack of complete understanding of this, there could have been some overlooked lack of precision. The rats that recovered due to stem cells were then examined through multiple tests to see what caused the recovery, and it helped shed new light on spinal cord injuries. This study could impact many of those that have suffered from irreversible spinal cord injuries, as it has shown potential treatment for the condition.
Thank you, Gager. I am Melody Franco, here to report the latest hard-hitting story. Today's breaking news: concerns for shorebird survival soars to new heights. Published on November 1st, 2017, a new study co-authored by the Wildlife Conservation Society reports concerns as Arctic adult shorebird populations are rapidly declining. Understanding the importance in monitoring and protecting the habitats in which these shorebirds breed and stop over is critical not only to their survival, but global migration. Anthropogenic factors minimizing biodiversity primarily include climate change, habitat loss, and invasive species. To quantify adult survival and the effects of anthropogenic factors, Dr. Rebecca Benson and her colleagues collected data across 9 breeding sites in the Canadian and Alaskan Arctic through the 6 species of shorebirds displayed. They tested the effects of environmental variables, study site, nest fate, and sex on annual survival rates of each species. They also predicted that survival rates of adult shorebirds would decrease with earlier spring snowmelt, warmer summer temperatures, higher abundance of predators, and lower abundance of arvicoline rodents. To further investigate, I traveled to the Arctic to talk firsthand with one of the researchers for the study. Roll the clip. Well, I have arrived on site in the Arctic. Finally made it, flew all this way. It is freezing, let me tell you, but we have found this fire to warm us for our interview. And I have a few questions for one of our researchers braving the snow.
Yes.
So, sir, this study found that adult survival rates for the shorebirds were higher than previously published for 5 out of the 6 shorebird species. So, do you see this changing in the future?
Yeah, well, you know, with the warming and the more variable climate added to the oil extraction occurring along the Chesapeake Lake in the National Petroleum Reserve, I would definitely conclude that our role in preserving Arctic habitats is still incomplete.
I would definitely agree with you.
Yes.
Additionally, the study found that the 5 species did not suffer from decreased adult survival, migrated from breeding sites in the Alaskan and Canadian Arctic to wintering areas in the Americas farther south. However, the dunlin species had decreased adult survival and migrated to wintering areas in the East Asian Australasian flyway on the west side of the Pacific.
Yes.
Why is this?
Well, you know, me and my buddies were just talking about this at dinner, and there's just a loss of habitat at the migratory stopovers or wintering sites in that area, which is decreasing adult survival for that species, thus making it a focus for future conservation efforts.
Yes, well,
Yeah.
Thank you for your time.
Looks like it's getting snow here pretty soon.
I hear it.
You know, in the Arctic,
Oh my goodness.
it really,
It's
kind of a direct snow. It's really localized.
This is crazy. Back to you
And,
in the studio.
Yeah.
Also found through this study was that survival rates were not affected by timing of snowmelt or summer temperature, and factors such as previous reproductive success, predator abundance, and the availability of alternative prey did not broadly affect survival. Additionally, this study has provided the first opportunity to research patterns in adult survival of Arctic breeding shorebirds within North America. Annual survivor estimates were relatively high, with variables, particularly sex and nest fate, not affecting survival rates. The local conditions also affected survival rates in only a few cases, suggesting that other conditions or variables are more influential that were not tested, such as food availability, habitat quality, or disease. With conditions in the Arctic continually changing, however, it is possible that the variables tested in the study will become more influential over time. The study concluded that environmental and ecological variables showed little effect on the annual survival rate of adult shorebirds at the Arctic breeding sites, but instead, migratory stopover and overwintering locations could be what is decreasing their survival rates, turning the direction of research for future studies to continue with further investigation in the future.
Hello there, Lucas Frankie here, and today we'll be talking about invasive species. Invasive species are non-native organisms that were introduced to a foreign ecosystem in one way or another. Due to their unnatural presence in the region, they are considered a threat to the stability of the ecosystem, and the clam Corbicula fluminera is no exception. These little mollusks recycle and release nutrients into the water, which contribute to algae growth. If left unchecked, the clams can seriously damage the quality of freshwater lakes where they are present. Luckily, scientists are working on a test to effectively monitor the presence of an organism in an ecosystem. This test is called an eDNA assay, and no, the E does not stand for electronic. I learned that the hard way. eDNA is short for environmental DNA, which refers to the DNA found present in an environmental sample, like water. Scientists obtained water samples from 11 different freshwater lakes in Nevada and California and analyzed them for the presence of Corbicula clams. Corbicula DNA was found in all 4 lakes where the clams were known to be present, and no DNA was found in the remaining 7 lakes where no clams were present. Based on these results, the researchers determined that the eDNA assay is an effective way to track organisms in an ecosystem. With this study, scientists hope to monitor the growth of invasive species and stop them before they have a chance to spread. I'm Lucas Frankie, and you're watching the Johnson Biology News Network.
New evidence suggests that the jellyfish Cassiopeia is capable of a quiescent state, which could give biologists a better understanding of where and how that sleep evolved. Okay. The study focused around Cassiopeia, which is categorized into Cnidaria, one of the first metazoan phyla to evolve tissue-level organization and differentiated cell types. The jellyfish has a non-centralized nervous system. Through the study, researchers aim to support the theory that sleep evolved before the emergence of the centralized nervous system. Typically, evidence of sleep capability has been found continuously in vertebrae, which have centralized nervous systems. In order to support that Cassiopeia has a quiescent state, which is a prerequisite for sleep-like states, the experiment consisted of a pulsation test and a responsiveness test, both comparing daytime versus nighttime for the jellyfish. For the first test to support a period of quiescence for Cassiopeia, pulsation was monitored for the jellyfish during the daytime and nighttime. Typically during the daytime, they had a consistent pulse rate of like 1 pulse per second, which you can see right there in diagram 1 and diagram A. Correspondingly, the inner pulse interval was found to be the highest during the day, peaking at the time of feeding, which you can see right there. And then you can see, on the other hand, that pulsation at night was irregular and definitely slower. Okay. All right, and then this is a responsiveness test. So the diagram illustrates that during the daytime, the first pulse after stimulation, so when the jellyfish was moved into open water, happened faster than the first pulsation at night. So basically, it's like jellyfish is inclined to be on surface levels. So it'll, like, if it's moved, it'll go straight down to surface level. So at nighttime, when when its pulsation was slower, it took longer for it to return back to the surface level. And then, but when it was in the daytime, it went straight to surface level. Okay. And then you can see like the time to first pulse and the time to the bottom for both like reaction in the daytime is clearly it took less time to the first pulse after being moved. And then the time to the bottom, it took longer for the nighttime jellyfish to react because it was less awake, which kind of proves that a quiescent state was existing. Because like in humans, like if you're woken up abruptly, it takes longer time to have reactivity because you're not as high functioning. Okay. All right. For humans, sleep acts as a homeostatically regulating function, a defining evolutionary purpose behind sleep. And in order to support that the quiescent state acted as a homeostatically regulating function for Cassiopeia, researchers continuously stimulated the jellyfish for 6-hour and 12-hour increments. So like you can see the nighttime here and there. So it was stimulated by like a swift like water flow was directed at it to keep it awake every 20 minutes. And since because of that, it was found to be 12% less active the next day when it was the 6-hour perturbed night, and then it was found to be 17% less active on a 12-hour perturbed night. And that's just telling you that the sleep was similar to humans and like vertebrate animals in the sense that the next day they'll be less reactive without the necessary sleep and like the lower pulsation time. So okay. And basically, in conclusion, this study largely supports the theory that a quiescent state exists in Cassiopeia, suggesting that sleep was evolved before the emergence of a centralized nervous system. And this study and like other ones that are similar to it can kind of give us more background into where certain the emergence of certain properties existed like in a time platform from like creation to now.
Yeah. Good morning and welcome to the Johnson Biology News Network. I am your host, Alfie Brownless, and today we'll be discussing a new experimental surgical technique used on rats. Love them or hate them, with their genome 80 to 90% homologous to humans, rats are critical in developing faster and more accurate solutions to human injuries and other problems. In an experiment run by a group of Brazilian scientists and approved by the ethics community for the care and use of experimental animals, an Achilles tendinotomy in rats was improved upon under light microscopy and sparing of the plantar's tendon. As one of the most frequent tendon injuries,
human Achilles tendon rupture frequency has increased in recent years, most notably resulting from the increased number of middle-aged and elderly athletes. Although many experimental models of Achilles tenotomy in rats have been developed and executed in a variety of different ways, little attention has been paid to the plantaris tendon. Being very developed and able to carry a substantial amount of weight in rats, the researchers from this study decided to focus upon it, developing an improved method for complete Achilles tenotomy. During the procedure, each rat was prepared with the aid of light microscopy under general anesthesia. In order to conduct the surgery, an assistant maintained the stability of the animal, incisions were made revealing the inner tendons of the organism, and, while carefully preserving the plantaris tendon, the Achilles tendon was severed. Following this, retraction of the proximal stump of the tendon was observed during passive foot dorsiflexion. Lastly, the skin inclusion was sewed with non-absorbable sutures and cleaned. After different time frames had passed, different groups of rats had their right Achilles tendon dissected, fixed in a 10% formaldehyde solution, and processed for paraffin inclusion. Then, 5 micrometer thick slices of tissue were cut and processed to histological studies to observe general tendon healing, especially regarding collagen and elastic fibers. In all cases, the animals fully recovered from anesthesia and exhibited a return to normal behavior following the procedures. All animals showed healing of the wound without skin necrosis. No drainage, discharge, or bleeding was observed until the 8th week after tenotomy. 2 weeks after the operations, the tendon rupture sites showed signs of healing, where the gelatinous connective tissue forming a bridge between the edges of the tendon and the picrocereus red technique under polarized light showed high amounts of red, thick fibrils, suggestive of type 1 collagen. At the 8th week, there was a shift to a higher amount of thinner collagen fibrils, slight yellow-green colored, likely type 3 collagen. These shifts in collagen types and levels reflect a normal healing process and allow for this surgery to be further deemed successful and stable. Since the 1930s, several studies have attempted to elucidate the process of tendon regeneration through experimental models of injury to the Achilles tendon, but there have been no standard surgical technique thus far to reproduce complete rupture of the Achilles tendon in rats. By isolating and preserving the plantaris tendon, the subjects in the study were much more easily able to heal after the procedure, allowing for future similar experiments to be conducted regarding Achilles tenotomy. With this stable, safer procedure, it will become much easier for future tests on this subject to be conducted that will be able to directly benefit humans.
Hello, America. This is Zach Johnson reporting live from the Johnson Biology News Network. There's good news in the biology world today, as laboratory researchers have discovered a vaccine that protects against meningococcal meningitis, a rare but debilitating disease that causes severe inflammation of the brain and spinal cord. According to the American Society of Microbiology, laboratory researchers tested the MEN-B-4C vaccine, also known by its commercial name as Bexsero, on the bacteria responsible for the disease known as Neisseria meningitis, shown right up here. The Bexsero vaccine was originally approved by the FDA in 2015, but has not undergone extensive testing in the medical field. The vaccine contains 4 antigens, shown here, which are proteins found on the surface of the bacteria Neisseria meningitis. These antigens elicit an immune response, releasing the antibodies necessary to counteract a bacterial infection. The Bexsero vaccine targets a specific strand of the Neisseria meningitis bacteria, known as serogroup B. This specific strand was responsible for 40% of meningococcal meningitis cases in individuals of all age groups, and 60% of cases in infants. During testing, the researchers tracked Bexsero activity using the meningococcal antigen typing system, or MATS for short. Using MATS, the researchers found that the vaccine protected against approximately 91% of the serogroup B samples overall, and more than half of the samples were covered by 2 or more antigens in the vaccine. The NHBA antigen, The NHB antigen, seen in the top right up there, covered 83% of the samples, which was the most universal of the 4. The FHBP antigen, in the top left, covered 53% of the samples. Pore A, in the bottom right, covered 5.9% of the samples, and NAD-A, in the bottom left, covered 2.5% of the samples. What does this mean for the future? Neisseria meningitis is a leading cause of bacterial meningitis in infants and young adults. With a new vaccine to counteract the bacteria, we may be able to protect against meningococcal meningitis, securing the lives of future generations. Yet another disease brought to its knees. Zach Johnson, Johnson Biology News Network, Midlothian, Virginia. Now, on to Jake Tenkata.
Thank you, Zach. Hello and welcome to the Johnson Biology News Network. I'm your anchor, Jake, and today we have a recent breakthrough in the medical field, as a recent study has found a new drug target for the silent but deadly acute myeloid leukemia, or AML. Leukemias are cancer that begin in the cells that develop into the critical blood cells we all need, while AML goes by other names such as acute myelocytic leukemia or acute myelogenous leukemia. Its progression and effects are the same. The word acute refers to the speed at which the cancer progresses, and if not identified within treated with a few months, it is often fatal. Myeloid is the specific cell in which the cancer originates. It first shows up in the myeloblasts, the immature form of the white blood cells, as shown by the dark purple shells behind me, which are plagued by cancer. These crucial parts are formed in the bone marrow, and in order to help those afflicted, it often requires a bone marrow transplant from a donor who matches you, often being a family member paired with chemotherapy. This type of treatment can be extremely debilitating and requires months of treatment and hospital recovery, for only a 30 to 40% chance of survival in people under 60 years old, and a 15 to 5% chance for people over 60. To find potential new forms of treating AML, researchers used CRISPR-Cas9 to perform gene editing. Introducing the Cas9 enzyme, it acts as a pair of molecular scissors to cut the specific gene out of the cancer-afflicted DNA. Paired with a guide RNA pre-designed by the researchers to interact with specific portions of the genome, they tested different mutations in mouse leukemia cells to target DNA essential to the survival of AML. Together they found 46 likely candidate genes. Among them, methyltransferase-like protein 3, or METL3 as I pronounce it, was found to react stronger to their testing. Essentially, for proteins to be produced into the cell, the DNA must be transcribed into RNA, which is then translated into the proteins required by AML cells. METL3 was an obvious choice, as it was required for the survival of AML cells, but had no effect on the creation of healthy blood cells. Connected to a total of 126 genes, several of which were critical genes, METL3 attached to methyl groups to the epigenome, which increased the ability of the RNA to be translated into proteins. By inhibiting this gene, no methyl groups were added, and therefore AML cells begin to die. Summarized best by Dr. George Vassallo from Cambridge University, AML is a feared disease that affects people from all over the world. Our treatment changed little in decades past, and outcomes remain poor for the majority of patients. We believe that we had to think differently and look in new places for ways to treat diseases, and in METL3 we have found an exciting new target for drugs. We hope that this discovery will lead to more effective treatments and that will improve the survival and quality of life for patients with AML.
Hi, my name is Lauren Bornstein with Johnson Biology News Network. In today's breaking news, we will be discussing individual-handedness in honeybees. A recent study at the University of Queensland in Brisbane, Australia, has provided evidence that honeybees, like people, can be either left-handed or right-handed. In order to test this hypothesis, honeybees were placed in a tunnel containing a barrier, as shown in the slide behind me. The tunnel had a consistent pattern on the inside so as to coordinate with the bee's natural centralized flying path. At the end of the tunnel was a feeder. In one test, the barrier had two apertures of different sizes, with the left choice being narrower than the right. In the second test, the barrier had two same-sized apertures. Overall, bees tended to choose the larger aperture at a rate of about 80%. This is most likely due to the bees wanting to decrease their risk of injury while en route to foraging, as shown in the graph behind me. The transit time, or the amount of time it took a bee to choose a route and fly through it, was longer when a bee chose the smaller aperture, taking further precautionary measures against injuries such as wing damage. This graph shows the varying transit times upon exit at different widths. However, across the board, transit times were significantly less when exiting than entering, meaning the bees were flying at a faster speed when returning to the hive. One reason for this pattern may be that as a foraging species, the hive is dependent on the worker bees' return. When the apertures were the same size, each was chosen by bees approximately 50% of the time. These bees were alone in the tunnel when data was taken so as to ensure that the choice was not affected by the choice of other bees, emphasizing individual bias. Furthermore, the probability of bees choosing the right-hand aperture or left-hand aperture when they were entering the tunnel for food versus exiting the tunnel after receiving food seemed to follow the same trend as the probability of the apertures having the same width. Overall, the results of this study support the idea that bees have an individual bias towards flying left or right. This variation is beneficial to honeybee populations because if 100% of the bees were unbiased, all of them would try to follow one route, causing something like a traffic jam.
Despite this individuality, a wider aperture is typically chosen in ideal conditions so that foraging becomes more efficient as it decreases transit time. Unlike most other animal populations, honeybees show varying behavioral lateralization, but only in forage-related routes. Despite honeybees being a social species, this study of 102 bees displays varying lateralization depending on specific behavior, situation, and conditions. Thank you for tuning into JBNN for this report.
Good morning and welcome to the Johnson Biology News Network. This is Nicole Vembers reporting from the Dental Journalist Division with an outstanding new discovery. A recent analysis of bacteria present in the mouth concluded that types of bacteria that lead to periodontal disease were also associated with a higher risk of esophageal cancer. This research was conducted by Dr. Jiyoung Ahn, an associate professor and director from population science at the NYU Langone Health Center in New York for cancer research. Esophageal cancer is the 8th most common cancer and the 6th leading cause of cancer death worldwide. This is due to the fact that the disease is often not discovered until it is at an advanced stage, causing 5-year survival rates to range from 15 to 25% worldwide. This is due to the fact that the disease is not often discovered until it is at an advanced stage. Dr. Ahn stated that esophageal cancer is a highly fatal cancer and there is an urgent need for new avenues of prevention, risk saturation, and early detection. Previous research has shown that the periodontal disease caused by certain oral microbiota has been associated with several types of cancer, including oral, head, and neck cancers. This study examined whether oral microbiota were associated with subsequent risk of esophageal aneosarcoma, EAC, or esophageal squamous cell sarcoma, ESCC. Looking at the roots of the disease and the bacteria associated with it, periodontitis is caused by the bacterial infection of gingivitis that worsens and causes the gums to pull away from the teeth and form spaces called pockets. In the beginning stages, the gums start to become red and swollen and bleed easily. As you can see, as the disease progresses, the gums and intermediate material between the teeth become infected and inflamed. The spaces between the teeth are formed by decaying gums, become extremely infected, and the body's immune system is unable to fight the bacteria as the plaque spreads and grows below the gum line. If not treated properly, the bones and gum tissue that support the teeth are destroyed and may eventually become loose and have to be removed. Furthermore, esophageal cancer occurs in the esophagus, a low hollow tube that runs from your throat to your stomach. Your esophagus helps move the food you swallow to the back of your throat to your stomach to be digested. The cancer usually begins in the cells that line the inside of the esophagus and can occur anywhere along it. Causes of the cancer may be attributed to tobacco, alcohol use, obesity, or particular nutritional habits like bad oral health. In Ahn's study, she along with her colleagues collected oral wash samples from 122,000 participants from 2 large cancer screening health studies. 10 years later, 106 of the participants developed esophageal cancer. The group then compared the oral microbiomes extracted from DNA in cancer-free cases to the sequenced oral wash samples. Certain bacterial types found were associated with a higher risk of esophageal cancer, like high levels of Tannerella forsythia related to EAC and Porphyromonas gingivalis related to ESCC, which are both species of bacteria that are linked with common gum disease, and noted. This study also showed that certain types of bacteria were not causes of the cancer, but preventers for it. For example, the Neisseria bacteria found were associated with lower risk and may have a protective effect that could be potentially examined in further research, and concluded that this study indicates that learning more about the role of oral microbiota may potentially lead to strategies to prevent esophageal cancer, or at least to identify it at an earlier stage, and the next step is to verify whether these bacteria could be used as predictive biomarkers. All in all, the study goes to show that good oral health, including brushing your teeth twice a day and attending dental visits every 6 months, is an important way to guard against periodontal disease and esophageal cancer, among many other possible oral infections. Yet another reason why good hygiene habits are good for your health. This has been Nicole Vembers with your Daily Biology News.
Hello, my name is Valerie LeBron, and thank you for tuning into Johnson Biology News Network. In the news today, biomedical and biotechnological fields are stunned for a group of ETH researchers from Switzerland have created a biocompatible ink for 3D printing. On December 1st, 2017, the group of researchers led by Professor Andre Suter, head of the Laboratory for Complex Materials, created the biocompatible ink by combining cultivated bacteria with a specialized hydrogel. The functional living ink, or Flint for short, can be created with many types of bacteria, especially those able to degrade toxins, synthesize vitamins, form cellulose, and conduct photosynthesis. The hydrogel in Flink, made from biocompatible hyaluronic acid, HA, K-carrageenan, HCA, and fumed silica, Fa, not only acts as a habitat to maintain bacterial life, but also allows the metabolic activities of the bacteria to be utilized by the 3D printing material or structure. By combining the mobilization of the bacteria, by controlling the mobilization of the bacteria, the hydrogel also increases the product yield of the metabolic reactions of the bacteria, acts as protection from external environments, and allows for bacterial growth. So this is the creation of Flint. And so basically there's bacteria, and it's combined with the hydrogel, and it creates the Flint. And it can be used for bioremediation and biomedical applications. In order to outline some of the various functions of the ink, the researchers introduced 2 versions of Flink, each with different bacterial strain. The first, using bacteria Pseudomonas putida embedded within it, was able to degrade a toxin called phenol, implying the potential for future bioremediation applications, especially within the chemistry field. The second, a version containing bacteria Asobacter psilonum within it, created a 3D matrix structure, enabling the formation of high-purity bacterial cellulose, which is often important for biomedical applications, due to its pain-relieving properties. However, the functionality is dependent on its rheological properties. Rheological properties are characteristics determining a substance's ability to flow or be deformed. One of these essential properties includes viscosity, which is determined by the components of the hydrogel. The ETH researchers needed to reach an ideal viscosity that would ensure the survival rate of the bacteria, while leaving the ink stable enough to create accurate and stable 3D structures. If the structure is too viscous, the bacteria's ability to move and grow will be inhibited. However, if it is not viscous enough, it cannot support multiple layers, leaving it unable to form a 3D shape. Image D over here shows Flint, and so basically here you can see the layering of Flint, and over there you can see how with more layers it becomes more stable. Furthermore, up to 4 different kinds of ink, all varying with bacteria within them, can be combined with within one structure to maximize the number of metabolic properties that a 3D printed biostructure can possess. With the creation of this biocompatible ink, scientists believe that medical uses in areas such as burn treatments are possible because the bacteria used for the ink is naturally occurring within the human body. However, some applications are just for entertainment purposes, such as the world's first living tattoo, which was created on December 6, 2017, and comprised solely of 3D printed bacteria. Thank you for tuning in, and now to Nor for the latest news.
Thank you, Valerie. Mark Twain is notable for many achievements, but few people are aware that one of his interests were the flies Epidra hyans thriving on the shores of Mono Lake. Mono Lake is hypersaline and has a pH of 10. Only algae, bacteria, and brine shrimp can survive in the extreme conditions there. The lake contains deposits of sodium bicarbonate, carbonate, and calcium carbonate that form tufa under the lake's surface. As the surface of the lake has lowered over time due to evaporation, the tufa have slowly become exposed. It is using these structures that the flies crawl under the lake. The flies have evolved in such a way that they do not get wet while under the lake. If they were to be exposed to the water, a film of minerals would dry on their body, reducing their water repellent capabilities in the future and leading to eventual death. To prevent wetness, the flies are coated in a waxy substance and are covered in small hairs called setae. The hairs trap a layer of air, creating an air bubble around the flies as they crawl into the lake. While underneath, the flies feed or lay eggs. The air bubble allows them to remain underwater for up to 15 minutes. To emerge from the lake, the flies either crawl back up the tufa or let go and float to the top. The lake has a high concentration of sodium carbonate, making it clingy. The flies must exert forces about 18 times their body weight in order to break the surface of the water. To help the flies escape, Hofmeister effects interacting with ions in the water generate a small negative charge at the water's surface. As the flies come closer to the surface of the water, their positively charged hairs attract the negatively charged ions in the water, encouraging the bubble to break and releasing the flies into the air. Having realized such a unique interaction between organism and environment, the research team that conducted this study hopes to use their findings to encourage others to continue to study remote locations on Earth.
Thank you, Olivia. Good morning, and welcome back to the Johnson Biology News Network. My name is Alex Tang, and today we're going to Australia for our next news report. In Australia, there lives a genus of spider called daisies. This spider species spans over a total of 14 species, but two of the species are less understood, the Dacis harmeri and the Dacis thorax. The Dacis thorax is recognized and differentiated from the other species in the Dacis genus by its conductor plate and its conductor tip, two parts of the spider's anatomy, both which are more slender than the rest of the species. This species is found on the small island of Upolu, one of the smallest known islands off of Australia. Dacis harmeri, however, is a lesser-known species and has only been observed in juvenile stages. 4 of these juveniles are kept in museums. 2 museums are in Germany, and 1 is in Western Australia. As of last week, however, there has been a new addition to the Dacis genus. This new species is called Dacis bob marley. This new species is named in honor of Jamaican reggae singer-songwriter Bob Marley. Dacis bob marley takes this unique name because of its equally unique habitat, which may come as unexpected. This spider is not the land-dwelling spider that we're all used to. Instead, this spider lives in the intertidal areas of the coast of Australia, or the coast that fluctuates between land and water depending on the tide. This high-tide, low-tide nature of Dacis bob marley's habitat is the reason for its name, because of Bob Marley's hit song High Tide or Low Tide. In order to survive in this strange area, Dacis bob marley have grown to create intricate silk air chambers in shells or corals during high tide, and during low tide they become invertebrate predators, where they explore the intertidal wildlife and debris searching for prey. These spiders were only discovered because of special circumstances. On the day of discovery, scientists observed these spiders when the tide was especially low, on a coral called brain coral. The discovery of both male and female spiders of this species occurred in Australia's Sunshine State, Queensland, near the Great Barrier Reef. This species has been described 150 years ago, but was not formally reported nor illustrated. To illustrate them, the species were placed in a 75% ethanol solution, dissected, and photographed digitally with a digital camera, and the model was assembled in Adobe Photoshop. The anatomy of the spiders were then labeled. The scientists proposed that the common name of this spider be Bob Marley's intertidal spider. At the moment, the exact distribution of the species in Australia remains unknown. This newfound species, named after reggae legend Bob Marley, may be the key to providing insight to further research into the lesser-known species in the Dacis genus. Now on to Drew for more biology news.
Thank you, Alex. This is Drew Ellison with the Johnson Biology News Network coming to you from Midlothian, Virginia. A group of researchers at Cornell University in the Department of Biological and Environmental Engineering has created a new, groundbreaking way for diabetes patients to receive insulin. Diabetes, involving a bodily inability to produce insulin, leads to an increase in blood sugar levels, which makes it imperative for these levels to be constantly monitored. There is no cure for diabetes, and over 1 million Americans must carefully manage the disease every day. There is no cure, In the case of type 1 diabetes, insulin cells are produced in pancreatic cell clusters called islets, but these cells are then destroyed by the body's immune system. The team at Cornell was able to discover a way to implant thousands of islet cells into the body at one time, reducing and potentially eliminating the need for shorter-term insulin regulators like shots or pumps. Calling their creation Traffic, an acronym for Thread-Reinforced Alginate Fiber for Islets Encapsulation, the Cornell students and professors joined together to make a seamless insulin implant. In terms of its construction, the islet cluster cells are compacted into a polymer thread and surrounded by a protective hydrogel coating such that the insulin will be administered according to the body's needs. There are no gaps between the hydrogel-coated capsules, and the thread is twisted into a porous helix shape so as to ensure a sturdy base for the insulin. Prior to this research accomplishment, professionals have thoroughly studied the concept of covering islet cell clusters in a hydrogel coating. However, these studies involve multiple clusters being transplanted separately instead of as a part of a polymer thread. The reasoning behind why this technique was ineffective is because the implanted cell clusters must be removed from the body once they are no longer needed or useful. If left in the body, there becomes a potential for tumor growth. Through the research team's method, however, it is easy for surgeons to place and remove the thread of islet clusters as needed due to their fixed location in the body. The threads being currently used by the research team are approximately 6 feet long but only require minimally invasive surgery to implant. While testing continues to occur, the Cornell team estimates that these insulin threads can last anywhere from 6 to 24 months in the body. This groundbreaking medical advancement marks an important stage in diabetes research. Although there is not a known cure for the disease, through advancements like Traffic, researchers are getting closer and closer to finding a long-term solution for millions of patients. This has been Drew Ellison with the Johnson Biology News. Thank you.
Good evening, America. This is one of your many anchors this evening, Jackson Berkey, here reporting to you live on a groundbreaking new discovery regarding the fat cells found in the body. It's been known for many years now that 2 types of fat are found in the human body: white adipose tissue and brown adipose tissue, and the white adipose tissue, the white fat, is what causes a lot of the dietary obesity and leads to type 2 diabetes and certain kinds of cancer, and is what stores energy in the human body. Whereas the brown adipose tissue is what consumes energy and generates heat due to the high percentage of mitochondria aiding in metabolic activity in adults. Now a team of researchers from Washington State University, China Agricultural University, and Shanghai Agricultural University have reported on the response of white fat when exposed to retinoic acid. The response being a chemical alteration in the adipose tissue that browns it to a beige fat. This is done after the acid binds to RAR or RXR nuclear receptors. This begins a series of chemical reactions that activates VEGF-A, a vascular growth factor, that in junction with the retinoic acid activates a P38 mitogen-activated protein kinase, causing a phosphorylation of the previously mentioned RAR receptor. With more acid in the now phosphorylated RAR receptor, PRDM16, a gene that regulates the production of brown adipocytes, is activated and creates brown adipocytes that bind with other white fats and creates a variant of the much healthier and less damaging brown adipose tissue. This research, along with other similar studies, could help to curb, if not eradicate, the epidemic that is obesity in America. Thank you for your time, and please enjoy as more stories are being brought to you.
Hello, and welcome to Johnson Biology News Network. I'm Sydney Bell, here to explore today's breaking news story, which puts the phrase, sick as a dog, to the ultimate test. The discovery of soluble parasite antigens in supernates in Babesia canis, a parasite that can cause anemia, could be used as a vaccine against virulent bee in dogs. These canines suffer virulent bee, which is also called the canis infection, evident in the condition of the dog's gums. As a result of this new vaccine, the amount of dogs diagnosed with canis infection will decrease and with further research and development may ultimately disappear. This image shows B. canis, which may be the future of vaccines for the man's best friend. An experiment where an isolated immunoprotective B. canis protein resulted in a doublet on the Western blot. Based on the information gathered by this experiment, a vaccine was created. This vaccine was created based on the full-length CBA protein and its protective immunity against the virulent bee in dogs. Once the vaccine was given to a test sample of dogs, the sample was protected against virulent challenge infection caused by limiting parasite proliferation. The vaccine resulted in the development of clinical signs being prevented as well as the canines being quote-unquote self-cured. I'm Sydney Bell, wishing everyone and their dogs a happy, non-rough Thursday morning. Back to you.
Good morning, and welcome to the Johnson Biology News Network. My name is Gabrielle Levon, and today we'll be reporting the latest breakthrough in biological science. the use of metformin to inhibit the development and promote the resensitization of treatment-resistant breast cancer. Multiple drug-resistant malignancy, MDR, is a serious issue facing numerous cancer patients throughout the world. Although the origin of multiple drug resistance within tumors is unknown, it can be prevalent at any stage of multiple cancers, especially in breast, lung, colon, and hematological cancers. MDR is commonly identified by the overexpression of drug efflux pumps of the ABC transporter family, such as MDR1, BCRP, and HIFL1 alpha, which are otherwise known as MDR markers. Clinical trials using ABC transporter inhibitors have been conducted, however have not been successful due to toxicities and their inability to counteract all methods of resistance. Therefore, scientists are still searching for a possible treatment. Metformin is an oral drug that improves insulin sensitivity, even within individuals with type 2 diabetes mellitus, a type of diabetes in which an individual can test of resistance to insulin. Recent analysis has shown that patients with type 2 diabetes mellitus, DM2, have a 31% decrease in the incidence of new cancers while taking metformin. Additionally, patients with DM2 in either lung, colorectal, or breast cancer taking metformin have reported additional survival benefits. To test the effects of metformin on cancer cells, scientists decided to conduct a series of experiments. The first experiment tested the effects of metformin on MCF7 breast cancer cells who were resistant to deoxyribonucleic. In this experiment, the reproduction of MCF7 cells were measured after a 48-hour period of exposure to metformin. The results of this experiment were that the metformin hindered the growth of MCF7 cells in a dose-dependent manner in both types of cells. In the next experiment, scientists attempted to discover the effects of metformin on just different states of estrogen receptor alpha, the status of which is used as a prognostic and predictive marker of cancer outcomes. Therefore, scientists exposed metformin to both estrogen receptor alpha positive and estrogen receptor alpha negative cells. The results were that metformin inhibited cell growth within all of the estrogen receptor alpha cells. However, metformin had a greater effect on the estrogen receptor alpha positive cells. Tamoxifen is a drug that has similar effects on the estrogen receptor alpha. Through experimentation utilizing both tamoxifen and metformin, the scientists found that the combination was more effective in impairing the growth of MCF7 parental cells, suggesting that metformin may be capable of hindering many cancer-promoting pathways within the cell. The scientists also examined the quantity of MDR markers between MCR7 sensitive and resistant cells after exposure to metformin. The results of their work showed that in sensitive cells, metformin increased HIFL alpha protein levels in a dose-dependent manner. In resistant cells, metformin reduced the levels of HIFL1 alpha and MDR1, also in a dose-dependent manner. Overall, the study revealed that metformin activity is context-dependent. When the MDR markers were tested within mice with leukemia cell lines and patient-derived breast cancer cells, the MDR1, HIFL1 alpha, and TFPI1 protein levels decreased. The final experiment performed examined if metformin could be used to prevent the development of MDR. Within this experiment, a group of MCF7 cells were pretreated in 100 microliters of metformin for 48 hours, and a group of MCF7 cells without pretreatment were used as the control group. Both groups were exposed to small amounts of deoxyribonucleic in an effort to build up resistance. The results of this experiment were that MCF7 cells with the metformin pretreatment decreased the HIFL1 alpha, MDR1, and BCRP levels and prevented TFPI1 increases normally seen within deoxyribonucleic-resistant cells, therefore proving that the pretreatment of metformin does inhibit the MDR-associated protein expression. The scientists believe that the beneficial results shown through experimentation, along with a low-cost oral delivery, minimal side effects, and few contradictions of metformin, prove that this drug can have a clinical role within the treatment of cancer. They hope to continue experimentation and perhaps begin clinical trials. We look forward to see what they will produce in the future. And now to Madeline with the latest news.
Thank you, Gabby. Welcome back, everyone. I am Madeline Tatum, reporting to you live from room 400. On January 12, 2018, Caltech research professor Carlos Lois uncovered the entire brain wiring diagram of the Drosophila fruit flies. Lois created his own method for tracing the communication between neurons, which he named TRACT, or Transneural Control of Transcription. Within the brain, neurons connect together in the synapse. This space is vital for the sending and receiving of signals between neurons. As neurons send electrical or chemical signals through the synapse, fluorescent proteins light up both the neuron that is sending the messages and the one receiving them. TRACT can trace these particular colors, thus creating the brain wiring diagram.
Arguably the best feature of TRACT is that researchers can observe the connections in real time through a living organism. Therefore, the researchers can observe the changes in neural firing as the fly moves and interacts with its surroundings. Scientists have observed neuronal connections as well as the differences between the brain wiring on the basis of development, sex, and genetic mutations. These changes are monitored on a daily basis in the same test flies. Prior to TRACT, examining such connections required intensive labor to create thin slices of the brain. Additionally, scientists could not observe the neural firings of live animals, so instantaneous changes of the neurons could not be detected easily. On top of observing neurons, TRACT can also genetically engineer the fruit flies. The researchers can prompt the production of proteins among the receiving neurons, giving them a specific function. Due to the techniques with the genetic encoding, TRACT is ideal for use in the Drosophila or zebrafish. In the future, Lois hopes to use TRACT in studies regarding mice as he progresses towards discovering communications between neurons in the human brain. Scientists believe that neurological conditions like autism is a result of a neuron's altered connections. Therefore, Lois believes TRACT could eventually create wiring diagrams of the human brain to observe the complex workings behind this and other diseases. Now here's Parker with the latest news story.
Thank you, Madeline. Hi, my name is Parker Hall with the Johnson Biology News Network. Today we'll be looking at the genome downsizing, physiological novelty, and the global dominance of angiosperms. Angiosperms are any flowering plant. Kevin A. Simonin and Aidan B. Roddy noticed that angiosperms are the dominant plant in most terrestrial ecosystems, but they are still considered to be an abnormal mystery to their evolutionary biology. It has been believed that these angiosperms originally arose to dominance through increasing the maximum potential photosynthesis in the whole plant carbon gain, which allows them to outcompete the ferns and gymnosperms, although the gymnosperms and ferns used to dominate. Simonin and Roddy have found evidence that genome downsizing resulted in a rapid growth of flowering plants. The first thing done was to collect the distribution of genome size among 393 land plant species. The branch length behind me is color coordinated with the ferns, gymnosperms, and angiosperms. The orange bars at the tips are scaled to the genome size for each of these species. The blue represents the angiosperm, and it is clearly indicated that the angiosperm's genome size is significantly smaller than the ferns and the gymnosperm's genome size. Once Simonin and Roddy discovered the significant difference in the genome size, it was found that species with smaller genomes have smaller but more numerous stomata and high leaf vein densities. Genome size explained between 31 and 54% of the interspecific variation in LG, DS, and DV. The analysis showed that each of these traits for the genome size across all species showed that shrinking the genome brings GSOP closer to the GS max, which facilitates faster rate of growth. In conclusion, angiosperms have the smallest genome and are growing the fastest, which led Simonin and Roddy to discover that the smaller the genome size, the faster the growth rate is. And now on to Callie.
Good morning, and welcome to Johnson Biology News Network. I'm Callie Yost, and I'll give you a little hint on today's story. Think baguettes, croissants, soufflés. Yes, today's story comes from France, where scientists have found a potential link between regular physical activity and the blood flow of patients with carotid artery disease. In this life-threatening illness, patients suffer from a buildup of plaques in the arteries carrying blood to their brain. As these carotid arteries become more and more narrow due to buildup, blood flow is altered and the patient suffers from thrombosis or clotting and a high risk of stroke. In fact, studying blood flow or rheology of those suffering from carotid artery disease was the basis behind the study. Rheological properties of blood and blood cells were measured, one of which was blood viscosity. Blood viscosity is the resistance of blood flow through the body, which is affected by the two rheological properties of red blood cells, aggregation and deformability. Red blood cell aggregation refers to the clumping of red blood cells, a reversible process. Red blood cell deformability refers to the red blood cells' ability to move themselves in the direction of blood flow. All three of these factors are worsened by the tightening of the carotid arteries as plaque builds up on their walls. 80 carotid artery disease patients, 15 of which already had a stroke or other episode, and 14 healthy controls were all tested for their blood viscosity, red blood cell aggregation, and red blood cell deformability, as well as other blood-related tests. Additionally, they were asked to respond to a survey regarding their physical activity. The results showed that patients with carotid artery disease have a higher blood viscosity than healthy individuals. Also, the red blood cell aggregation had a positive correlation with the percentage of stenosis, or narrowing of the carotid artery. However, the red blood cell deformability did not change within the groups. In patients with a high risk of stroke, it was shown that regular physical activity decreased red blood cell aggregates, as shown behind me, and their strength. With less and weaker red blood cell aggregates, the blood flows through the body more normally, potentially lowering the risk of stroke even in chronic conditions. I'm Callie from JBNN signing off and reminding our viewers to get in their exercise for the day. You never know when it just might save your life.